Howdy, my name is Adamari Cerda, and my partner Mario Flores Castro and I will be discussing our fourth experimental lab report on the carbohydrates metabolic tests. To begin, the carbohydrates metabolic tests that this experiment focuses on serve as a way to further characterize bacteria. These tests ultimately examine a bacteria's ability to break down certain carbohydrates through events such as oxidation, fermentation, and the production of gas. This lab report focuses on identifying and understanding the biochemical pathways that enable bacterial identification. The oxidatively slash fermentatively glucose medium test for a bacteria's atmospheric requirements. It's a semi solid medium that uses pH changes to identify obligate aerobes and facultative anaerobes. The triple sugar iron agar tests serve to identify bacteria that attack certain carbohydrates within the medium through the production of gas and the possible presence of hydrogen sulfide. Lastly, the carbohydrate fermentation tests a bacteria's ability to degrade or ferment certain carbohydrates present within the medium through the production of acid or acid with gas. To perform the oxidatively or fermentatively glucose test, the bacteria needed are P. aeruginosa, E. coli, A. faecalis, and the unknown samples 101 and 201. For performing the triple sugar agar test, the bacteria needed are A. faecalis, E. coli, S. typhimurium, S. flexneri, S. aureus, P. mirabilis, C. ferendi, and the unknown samples 101 and 201. Lastly, the bacteria needed for the carbohydrate fermentation tests are E. coli, S. aureus, and A. faecalis, in addition to the unknown samples 101 and 201 for the glucose broth. For the lactose broth, the bacteria needed are E. coli, A. faecalis, and the unknown samples 101 and 201. Lastly, the sucrose broths will be needing E. coli, A. faecalis, and the unknown samples 101 and 201. When it comes to procedures, it is important to note that these steps will be applied to all mediums with respect to each bacteria that will be inoculated. When it comes to performing the oxidatively or fermentatively glucose test, we will be using a needle to inoculate the medium with a bacterial colony by stabbing through the medium 1/4 inch of the bottom for each of the two tubes. Label one of the tubes closed and cover the medium with 2 mLs of mineral oil. Label the second tube open and do not modify. Lastly, both tubes will be capped loosely and incubated for a few days. For the triple sugar iron agar test, we will be using a needle to inoculate the TSI tube with a picked up bacterial colony by stabbing through to the bottom of the medium and then streaking the surface of the agar slant. Secondly, we will cap loosely and inoculate for a few days. The last step will be to examine and record results. Lastly, the carbohydrate fermentation tests call for using a loop to inoculate the medium and a single bacterial colony. The tubes will be capped loosely and incubated for a few days before examining and recording results. The oxidatively or fermentatively glucose tests for Pseudomonas aeruginosa displayed no change as there were no color differences nor any growth that might indicate oxidation or the presence of a facultative anaerobe. Therefore, Pseudomonas aeruginosa was interpreted to be saccharolytic in both open and closed tubes. The oxidatively or fermentatively glucose test for Escherichia coli displayed a golden yellow color change in both open and closed tubes. Therefore, E. coli was interpreted as a facultative anaerobe. The oxidatively or fermentatively glucose test for the unknown sample 101 displayed no changes in both open and closed tubes. Therefore, this organism was interpreted as saccharolytic. The oxidatively or fermentatively glucose test for the unknown sample 101 displayed no changes in both open and closed tubes. Therefore, this organism was interpreted as saccharolytic. As for the oxidatively or fermentatively glucose test for sample 201, there were displayed color changes from the original green to a golden yellow. Therefore, unknown 201 was interpreted to be a facultative anaerobe. Lastly, the oxidatively or fermentatively glucose test for Alkaligenes faecalis displayed no color changes within the open and closed tubes. Therefore, this organism was interpreted to be saccharolytic as well. The triple sugar agar test for S. aureus displayed no change in color and was interpreted as a saccharolytic bacteria that is able to use glucose, lactose, and or sucrose for fermentation. The E. coli sample, however, displayed a red slant color with a yellow and slightly orange butt. This was interpreted as the organism's ability to only use glucose for fermentation.
The triple sugar agar test for S. flexneri showed a red slant and a yellow bud. This is because S. flexneri is only able to use glucose for fermentation. The results for A. mirabilis showed a yellow slant and a yellow bud. This is because it is able to use glucose, lactose, and sucrose for fermentation. The triple sugar agar test for C. friondi showed a yellow slant and a yellow bud. This is because C. friondi is able to use glucose, lactose, and or sucrose for fermentation. On the other hand, A. faecalis shows a red slant and a yellow bud, and there's a presence of black precipitation. The red and yellow are explained by the bacteria only being able to use glucose for fermentation, while the red while the black precipitation is explained by the bacteria able to produce sulfate gas. The triple sugar agar test for S. diffimurium shows a red slant and a yellow bud. This is because S. diffimurium is only able to use glucose for fermentation. For unknown 101, we also saw a red slant and a yellow bud. Again, this is because it is only able to use glucose for fermentation. On the other hand, unknown 201 has a yellow slant and a yellow bud. That means that unknown 201 is able to use glucose, lactose, and or sucrose for fermentation. This test tube also showed small cracks in the agar, which we interpreted as bubbles, which means there was gas formation. For the sucrose broth test, we saw how E. coli and A. faecalis they have a red broth, which means that these bacteria do not ferment sucrose. Unknown 101 has yellow broth and there's no gas present, which means that there's fermentation but there's no production of gas. On the other hand, unknown 201, there's gas present and the broth is yellow, which means that this bacteria is able to ferment sucrose and creates gas out of it. For the glucose broth test, we saw how E. coli shows a clear yellow broth. This shows that E. coli fermented glucose. S. aureus also shows a yellow broth, but there's more turbidity, shows that it's able to ferment glucose as well. Uno 101 also shows a yellow broth, which again means that it fermented the glucose. A. faecalis shows a red broth, which means that this solution is alkaline, so there's no fermentation happening. Unknown 201 shows a yellow broth, which means that it was able to ferment the glucose. For the lactose broth test, this is how E. coli has a yellow broth with no gas. It means that E. coli was able to ferment lactose. A. faecalis shows a yellow broth, so it means that it was able to ferment lactose. It also shows the presence of gas formation. Unknown 101 has a yellow broth, which means it fermented the lactose. Unknown 201 also has a yellow broth. There's also little to none gas, as we only saw one small bubble. The oxidative fermentation glucose test determines the atmospheric requirements of bacteria. The bacteria are tested on their ability to oxidize glucose aerobically, which means that it uses oxygen, or ferment it anaerobically. The triple sugar iron agar test tests a bacteria's ability to ferment sugars like glucose to the formation of gas, in addition to the production of hydrogen sulfide. Carbohydrate broths help characterize a bacteria based on the fermentation patterns, and detects acid or gas production by the fermentation process. Thank you.